Okie dokie. So we are finishing up um, chapter 5. It's the second part that we're covering, but it's the first part of the chapter physically, right? It comes first. Um, I mentioned in the last video why I like to do it this way. But essentially, what we're talking about in this week is we're talking about um, how cells divide, but more importantly, like why. And there's two different types of cell division that happen for different reasons. Okay. And so that is our focus, right? How you get new cells, basically. Okay. So, first of all, let's talk about different types of cell division. Okay. So, there are a lot of different reasons why cells divide. Okay. One reason is replacement, right? Because they're damaged or because cells get old, right? So depending on, the particular organ or part of the body or whatever, right? Cells wear out in a lot of places in the body. Okay. And so they need to be replaced when they get old and they die. Okay. Or let's say you have a boo-boo, right? You have an injury, you need to replace cells. Okay. Um, Another function for cell division is growth, right? So every single one of you sitting watching this video, um, you all started off as a single cell called a zygote, okay? And now you're sitting wherever you're sitting, and you are made of approximately 10 trillion cells, okay? How do you get that many cells? Lots of cell division, okay? So this is showing a very early human embryo that is only 4 cells, okay? So lots of cell division. Um, some organisms, well, let me back up and not say that, okay? Organisms also use cell division as a means of reproduction. Some organisms do something called asexual reproduction, and other organisms do something called sexual reproduction. We've talked about this a little bit, but we're going to kind of get into the differences, in this chapter. Okay, so some organisms, lots of unicellular organisms, in fact, the majority, I shouldn't say all, I should say the vast majority of unicellular organisms, the way that they reproduce is simply through the first type of cell division that we're going to talk about. Okay. Um, Remember that asexual, right? When you put why is my pen not working? There we go. When you put a in front of a word, it means without. Okay. So without sex, meaning you don't have a partner. Okay. You just replicate your cells essentially. Okay. So here's an amoeba doing asexual, right, reproduction by doing mitosis, a type of cell division. The thing that's interesting and I think surprising to lots of people is that there are also a lot of multicellular organisms that can do that only do asexual reproduction or can do both asexual and sexual. Okay, so these two examples here, sea stars and a lot of plants, specifically this is showing you an African violet, but we'll look at other examples too, can perform both types of reproduction. Okay, and they're beneficial at different times. Okay, so asexual reproduction is most useful if you're unicellular or if there are complications with finding a partner, right? Makes it more difficult. Or if conditions are great and, things are going well for you, just, copy yourself, do that. Okay. In the case of sea stars, it's kind of a unique situation. So with sea stars, the circumstances that allow them to do asexual reproduction are damage. Okay. So if you damage a sea star, this is one of my favorite, like, haha, you should have, learned more in school kind of, snob jokes. When I was working at the fish market in Massachusetts, where I worked in college, there were these fishermen that were pulling sea stars out of the ocean on their fishing boat, and they were using a trawl, which is a fishing method that scrapes the bottom of the ocean, and so they're pulling all this stuff out, and they pulled out a bunch of sea stars, um, but they can't sell those for anything. Like they're not valuable to them, right? That's not what they were fishing for. They call it bycatch. Okay. And what they were doing is they were sitting there and they were breaking them apart and throwing them back in the water, right? Thought they were killing them. When in reality, what they were actually doing is they were doubling the number of sea stars. Because if a sea star is broken in half or even in smaller pieces, as long as the broken piece contains part of this central disc that I kind of colored in red, right? So if it breaks right here, okay, it can regrow the missing parts. So you can break a sea star in half and end up with two sea stars, or break off an arm, and that arm, that's what's happening here, will grow more arms. Yeah, here's another picture of an arm that broke off that just, regrew the missing parts. The other piece regrew the missing arm. There you go. Right now you have two sea stars that are clones instead of just one. Okay. So asexual reproduction, right? Many plants you can grow by asexual reproduction. So if you've ever taken a cutting of a plant, that's asexual reproduction, right? You take a piece, stick it in the ground, right? Comes from another plant, did not involve any sex. Okay. A lot of plants have structures that are intentionally for this purpose. So here is a strawberry plant. Strawberry, if you've ever tried to grow your own strawberries, it's pretty fun. What strawberry plants do is they make these long little runners, right? So you see this stem here that's kind of like going along the top of the ground. And then at the end of these little runners, they basically produce a little baby plant that is a clone, a genetic clone of the parent plant, but it's a little ways away. And that little guy grows roots. You can see the roots down here, and it has its own leaves and its own stem, and you get a new strawberry plant, right? Asexual reproduction. Okay. So that is fairly common. Okay. Now, sexual reproduction, we're going to focus on more in the second half of this chapter, because sexual reproduction involves a special kind of cell division called meiosis. Okay. So what we're going to be focusing on here in the beginning is mitosis, which is one type of cell division. Later on, we're going to talk about meiosis. So we're going to get to sexual reproduction when we talk about meiosis, okay? Sorry, child-related interruption. What else is new? Okay, so because we're talking about mitosis first, right? We're going to talk about asexual reproduction a little bit. When we talk about meiosis, we're going to come back and talk about sexual reproduction, okay? Okay. So the first thing to realize, right, is that the approach that we're taking here is we're learning about cell division in eukaryotic cells. We will not be learning about cell division in prokaryotic cells. It's actually simpler. And in some ways, you're probably like, yeah, I wish we would do that instead, but we're not gonna. Okay. Prokaryotic cells have different cell division for a variety of reasons. First of all, remember, prokaryotic cells don't even have a nucleus, right? Second of all, the shape of their DNA is different. Okay. So while all DNA is composed of a double helix, right? Here's a little segment of double helix. In bacteria and archaea, that double helix of DNA is arranged in a circle. Now it's a big circle, in not big, relative to us, but relative to the size of the cell, right? It's a single large loop. Okay. Yeah, so it's literally just like this circle of DNA, and we call it their chromosome, and they only have the one. Sometimes little loops pinch out. That's a different issue. We're not going to get into that right now. We will talk about that a little later on in the semester. Okay. Whereas eukaryotic cells, instead of having one big circular chromosome, eukaryotic cells have a number of strands that we call chromosomes. Okay. How many chromosomes? Totally depends on what kind of an organism you are. So in humans, we have 46. That is the standard number of chromosomes that we have. One thing that's really, really interesting is the number of chromosomes within eukaryotes varies pretty widely, right? And so here are a few examples from your book. My favorite one is bananas. bananas have something like 126 chromosomes, right? That doesn't necessarily mean that they're better or fancier or whatever. You might be thinking, but humans are so complex and sophisticated. Why do we have fewer chromosomes than a dog, right? It's just how many segments of DNA that you have, right? It doesn't necessarily mean anything about like how many genes there are or anything else, okay? So when we are looking at examples of cell division in this book, and when we're talking about chromosomes, we're going to be talking about there being 46 chromosomes because we're focusing on humans, right? Because we are humans, so we're interested in humans, okay? But what do I want to say? Yeah. Okay. Just realize that it varies, right? So some other organisms have different number. Okay. All right. So here we go, right? Here is a human, and we're showing you a cell from a human. There's a cell, there's our nucleus in the middle. You recall from when we were learning about cell anatomy that the nucleus is where the DNA is housed inside of a cell. So the number of segments of DNA, right? The number of strands of DNA or chromosomes that you have is 46. Okay. And this is an image of what they look like. Okay. So we often see them represented as like a fat little X, right? So there's a single chromosome. If you counted this carefully, there should be 46. Okay. Now, the other thing to keep in mind, and we talked about this with genetics a little bit, is that remember that you've inherited half of those from your mother and half of those from your father, right? So while you have 46 chromosomes, right, you have how many homologous pairs? Right? So you have 23 homologous pairs, right? And so we'll get into that again. We kind of talked about it in the genetics section. Okay, we're going to talk about it again when we get to meiosis. Okay, all right. One thing that's really, I think, worth remembering is that the amount of DNA that differs between humans is so small, right? So small. So the vast majority of our DNA, if we compare one person to another person, is identical. That's the stuff that's like, okay, this is how you build a human. Okay. And then the differences are the things that are different, right? So between these two young women, right, their skin color, their hair texture, their facial features, right? Perhaps their height. I can't tell in this picture, right? Those kinds of things that are, that appear different, and also other, sort of physiological things that are different. But it's really, really minimal. Okay. So the vast majority of the DNA that is in your cells, right, is absolutely identical to any other human. All right. And for that matter, the majority of it is the same as other apes, right? So, you may recall, you may not, but if you think all the way back to the macroevolution section way over at the beginning of the semester, one of the things we talked about is how humans and chimpanzees share 95% of their DNA. Yeah. So, there's a lot of similarities between different organisms, okay, as far as their DNA is concerned. All right. Okay, so with regards to DNA, we are going to talk more. We kind of started to talk about how DNA is a double helix when we were talking about macromolecules. Okay, kind of. We are going to talk more about it when we talk about how DNA is used when we're talking about protein synthesis. Okay, so that's all coming up. For now, though, right, we're going to talk about two different, I want to say, formats of DNA, two different forms of DNA. Okay, so most of the time in your cells or in any cell, any eukaryotic cell, okay, most of the time the DNA strands, right, and remember how many strands are there in humans? 46. Good. Okay. So most of the time, your strands are all kind of stretched out. Okay, so each chromosome, right, is this long stretched out strand. Now, the format of that long, loose strand we call chromatin. Okay, so there's still 46 pieces, they're just long. Okay, those long strands of DNA can coil up into short, fat strands, which we call chromosomes. Okay. Okay. And so this is showing you sort of how that happens. So the chromatin is the DNA strand, and then these little balls of protein, and it wraps around and it wraps and it wraps and it wraps until it makes a fat little X looking thing that looks like this. Okay. Um, so that's a little bit of terminology. All right. Why do that? Why is it different? Why doesn't DNA just stay in the same form all the time? Well, imagine if you were trying to separate a bowl of spaghetti and you had 46 strands of spaghetti, cooked spaghetti or ramen or whatever noodle you would like it to be. Okay. So you have 46 long, skinny noodles and you need to separate them, and you need to do it carefully because you want to have exactly 23 specific chromosomes on one side, and you want to have exactly 23 specific noodles on the other side. And you don't want them to be breaking. You don't want to get it all tangled up, right? Well, how are you going to do that? Okay. And you have to do it fast too, reasonably fast, right? So how do you do that? Well, if you've ever eaten noodles, you know there are different approaches to eating noodles. Okay. Um, so one way, one spaghetti trick, right, is you take your fork and you twirl your spaghetti, right? So you twirl the noodles onto your fork before you shove it in your mouth, right? Another way is to just slurp. Okay. Slurp makes kind of a mess though, and you're more likely to break off pieces, right? So while fine for eating, not so good for cell division. Okay. Because remember, we're trying to keep it neat and not break off any pieces. Okay. So when cells divide, you have to coil up your noodles before you move them just to keep everything tidier. So think of this as like you're making a little tidier package, right, of this long noodly thing in order to be able to move it around, which is fundamentally what's happening in cell division. Okay. So here is an image that shows us what a cell looks like when the DNA is still in that chromatin form or uncondensed chromosomes, right? So chromatin. Um, when it coils up, it looks different. So see how this inside just kind of looks like, I don't know, it just kind of all looks like homogeneous pink. And you see how here it looks like dark little purple squiggles. Now the color is completely artificial. They're using stain on these slides to, on these cells to make them look like that. But right, we go from this like completely even looking mass to there's like distinct squiggles. So I want you to help you understand that. I want you to imagine that you're standing on the top of a building, a tall building. Let's say it's 10 stories. I don't know. Okay. And you're looking down at a bowl of spaghetti. Well, you're not going to be able to see individual noodles, right? You might be able to identify, oh, it looks like there's some spaghetti in that bowl down there, right? But you won't be able to like pick out individual noodles. Okay. That's what this is, right? The noodles are so skinny. The strands of DNA are so skinny, and there's so many of them all crammed together that you can't like pick them out. So they kind of have this appearance. Once they coil up though, right, a little space shows up between them because they're all coiled up and it's a little bit tighter. Then you can start to see them under a microscope, see individual chromosomes now. Okay. So I would say this is condensed chromosomes as opposed to chromatin, which is in this picture. Okay. All right. Um, what do I want to say? Okay. Another thing you need to know is you need to know a little bit about what is a chromosome anyway. Often in your book, the way that you see a chromosome is as this kind of X-shaped thing. Okay. Now, the colors here are not just because they're trying to make it look all like galactic and cute, right? This is an actual chromosome that has been stained so that you can see different regions of it. Okay. So the colors aren't just, for fun, right? They're meaningful. Um, One important thing to realize is that chromosomes duplicate. Okay. And so when they do that, they go from a single strand to looking like a fat um, X that's connected in the middle. Okay. And that middle portion is called a centromere. I don't really care if you know that name, but that's essentially what's holding the copies together. Okay. So what I want you to think about is I want you to imagine that DNA essentially it's the instruction manual for a cell. Okay. And so if you are going to make a whole new cell, you're going to need complete instructions, right? You can't just like lose half of the instructions, right? You're going to need to have a complete set of instructions. And so if were an office manager and you were putting together, all of the documents for how to run your business, um, and you were going to make a new office, right? You were setting up a new office. You would probably want to make a copy of all of your, procedures, right? A complete copy of that before somebody moved into the new office, right? That's what chromosome duplication is about. Okay. So we replicate or duplicate the DNA so that now we have two copies of everything. So that when we ultimately, because remember, the point of cell division is to make new cells. When we ultimately have new cells, each new cell still has a full set of instructions. Okay. All right. We're going to get into the details of this. Okay. So just as a reminder, right, this is a picture of a duplicated chromosome, right, that is composed of two strands, and we call them sister chromatids. It sounds an awful lot like chromatin, which is kind of irritating, but I don't know what to tell you. I didn't make these names. Okay. And sister chromatids are the copies, right? So sister chromatids, what they are is this side, right, is an identical copy of this side. And those copies are held together until it is time to separate them. Okay. All right. So that's what this like fat X is. And like I said, the centromere is the middle area that holds them together. And oh, look, they're pointing to a specific gene, right? Remember, a gene is an area on a chromosome that has instructions for some kind of trait. Yeah. Okay. All right. So one more picture, and then we're going to get into the actual cycle. Okay. So here is an image that shows an area of a plant that is actively growing. This happens to be the root tip of an onion. It doesn't really matter, but there it is. Okay. And so what's cool about this image is what you're seeing like a moment in time, like captured, where they have a bunch of different cells here that are in various different stages of this process of cell division. And we're going to talk about the stages in a moment. So you can come back to this and try to figure out what all you're looking at. Okay. But the thing that I want to point out, first of all, is look at this chromatin, uncondensed chromosomes, right? It just kind of looks speckly. You can't see individual chunks. But then by the time we get to like these periods in time, see how it looks like squiggly, fat little wormy things? That's that condensed DNA, okay? Condensed chromosomes. Okay, so the cell cycle. Essentially, you can kind of think of this as the life cycle of a cell, all right? Now, the first thing I want to say is that the life cycle of a cell is not the same for all cells, right? This is the process by which you get new cells. Okay, this is how you get new cells, whether you're replacing them because of damage or they're old, or for growth if you're just trying to grow, or right, or if you're asexually reproducing, right? This is the way that we get new cells. How quickly this happens, how often it happens, whether it happens at all, varies quite a bit actually, not just from one organism to another, But even within a single organism, it varies, right? So I'm thinking of humans as an example. Imagine that convenient example. Your skin heals incredibly well, right? So, if you get a boo-boo, right? Generally, depending on how severe it is, but generally within a few days, right? It's completely, the damaged cells are replaced. Okay. Sometimes you need to help it along by like stitching the skin together so that it doesn't stay open, right? You kind of hold it together, or sometimes they use glue. I love medical super glue. It's the best. That way you don't have to get stitches. Just glue that sucker together. It's awesome. Right. So, you know, that happens pretty quickly, right? You're making replacement cells relatively quickly. Okay. But there are some cells in your body that replicate at some point in your life, but those cells stop dividing and they no longer divide again after a certain point. So neurons are like that, right? You don't make new neurons. By the time you're born, you basically have, there's some exceptions to this, but for the most part, by the time you're born, you have all the neurons you're ever going to have. This is one of the reasons why I'm like, don't do drugs, kids, right? Because if you if you kill the neurons that you have, you can't replace them, and you have a limited number, right? And we lose them anyway due to wear and tear, right? So don't increase the wear and tear for fun. Just don't go there. don't do it. Don't do it. Okay. All right. So the speed and whether or not this happens varies. Okay. That was my punchline there. All right. The cell cycle basically is two major phases, and then there's like a breakdown of smaller phases. Okay. So most of a cell's life is spent in interphase. Okay. So during interphase, a cell is just, it's doing its thing. It's doing what it does, right? It's doing its normal cell activities. Okay. In the beginning part of interphase, you have a cell that has some number of chromosomes, right? So if, we're talking about a human cell, it's going to be 46, right? Because we already talked about that. In this example, they're trying to make a clean picture. So they're showing, right? What they're showing you is they're saying, oh, they're just showing you one, right? To keep it simple. Okay. All right. So it grows and it's doing its normal, it's doing its normal day-to-day functions. Okay. Keep on cruising. Then kind of in the middle-ish of interphase, right? In the middle. The chromosomes get duplicated. Okay. So let me go back to this picture, right? So this duplication where each side of the chromosome is copied. Well, I shouldn't say each side because it starts off as one thing. So when the chromosome is copied so that you end up with two sister chromatids, right? That happens in interphase, okay? So you start off with a single strand and each of those strands is duplicated in interphase, right? So that's what they're trying to show you here. They're trying to say, oh, look, we took that single chromosome and we duplicated it. Okay. Now remember that actually those two copies are held together until it's time to separate them. Okay. So DNA is replicated. We sometimes use the term replicated or sometimes we say duplicated. Same difference in interphase. Okay. So when the cell is doing its normal stuff, well before the actual dividing starts. Okay. So then this stretch of time here, they're calling the mitotic phase, or we can just call it mitosis. Okay. Now you'll also see another word, cytokinesis. We'll get to that in just a second. Okay. So mitosis is the actual process of the DNA separating. Okay. So mitosis, right? DNA separating. Right. And so remember, we made a copy, right? We made all of our copies back in interphase. So now we can separate our copies. Okay, so that's what happens in mitosis. And, mitosis makes people crazy because, it's a process that seems kind of foreign. I do want you to know what's happening in the steps of mitosis. That's going to take a little bit of practice maybe, okay? I don't know, okay? I want you to understand mitosis in a pretty straightforward kind of way. Okay, so let's look at some images. All right, so now that we know kind of the order of things, right? We have this interphase, which lasts typically a really long time, and then we have mitosis. Let's look at some images. So this is the first, this image represents the beginning of interphase. So before the duplication happens, right? So, oh, there's a nucleus. I mean, it would be inside of a cell. So they left out the rest of the cell because they didn't care, right? So we have a nucleus and it has some chromosomes in it. Now, each of these squiggly lines represents a single chromosome. How many chromosomes do we have here? 1, 2, 3, 4, 5, 6. So this is clearly not a human cell, because if it was a human cell, it would have 46, right? So the reason they're not showing you something with 46 in it is because that it gets overwhelming real fast if you've got more than, 6 or 8 to look at. Okay, so we've got our chromosomes here. They are all in that uncondensed chromatin state. Okay, so they look like little strands. So that's the beginning of interphase. Well, remember in the middle of interphase, those get duplicated. And so this is kind of showing that process of duplication happening. You don't need to know that process right now. We're going to talk a little bit about it later though. Okay, so we'll get to that. So now what we're looking at is so once again, there's our nucleus, right? And you can see here, 1, 2, 3, 4, 5, 6 are 6 chromosomes, chromatin strands, right, have been duplicated. Okay, so now we have, each one has the two sister chromatids that are attached at that little middle part. Okay, they are still not condensed. Okay, so they are still in that long, lanky chromatin form. They are not condensed yet. Okay, so that's like the middle, I should say middle to end of interphase. Looks like this, right? Because this happens in the middle of interphase-ish. Okay, all right. Then after interphase, if we go back to our cycle, right? Interphase is all of the orange. So interphase leads us into mitosis, the mitotic phase. So let's look at some of those. Okay, so prophase, this is the first step of mitosis. Woo! Woohoo! Okay. All right, what is happening here? There are some things happening. Okay, so, right, remember that purple circle represents our nucleus. Okay, there are actually 3 distinct things that all happen during prophase, during this first step, that are significant. Okay, so one of them is you can see that the nuclear membrane, the nuclear envelope, is starting to break down, it's starting to dissolve. Why is it doing that? Well, remember that we're going to be separating our DNA here, and so it can't still be trapped inside of a nucleus if we're going to separate it into two piles. Okay, so the nuclear envelope kind of breaks down and dissolves. All right, another thing that happens is those chromosomes condense. So up until this point, right, we've been talking about these long, lanky chromatin style chromosomes, but they're condensing down, right? And they getting chunky, okay? And remember, they were duplicated way back in interphases when they were replicated, right? So these fat little chromosomes will look like X's because the two sister chromatids are the two copies. Okay, how many chromosomes do we have? Still the same number. So we started with 6, we duplicated them, so now we have 6, pairs of sister chromatids, and now we have 6 fat little pairs of sister chromatids. Okay, so we still have 6 chromosomes. All right, and then the last part, and this is happening outside of the nucleus. So parts 1 and 2 are happening inside of the nucleus. The third part is the development of something called the mitotic spindle. And what the mitotic spindle is the mitotic spindle is parts of cytoskeleton. So perhaps you remember that when we were talking about the all the different organelles and structures in a cell, we talked about how the cytoskeleton is responsible for for supporting the cell and also for movement in the cell. Well, the mitotic spindle is this specialized cytoskeleton whose job it will be to move our chromosomes around. Okay, so we're going to see the mitotic spindle in action in a little while. Okay, but for now, it just kind of looks like a cage, right? That is formed. So it's all of this like orange and yellow, all of this, right, is the mitotic spindle. And what it's going to do is it's grabbing onto those chromosomes, and it's going to ultimately be responsible for separating the sister chromatids. Remember, they're copies, right? Now, here's a picture of what this actually looks like underneath a microscope. So obviously the other two pictures are all, illustrations. The illustrations keep getting more and more realistic as I age. Right, the illustrations get better and better, but here's an actual picture. So keep in mind that this picture over here is showing 6 chromosomes, and most cells have way more than 6, right? So in reality, it's actually pretty complex because we're dealing with a larger number of chromosomes than just 6 most of the time. Okay. So that's prophase. So prophase is by far the most complicated of the phases because 3 different things are happening. The nuclear envelope is breaking down. The chromosomes are getting condensed. They had already been duplicated way back in interphase, right? But they're getting condensed. And then the third thing is the mitotic spindle is forming, right? And that's cytoskeleton that will move sister chromatids. I didn't finish my thought. Okay, so those are the 3 things, and they're all happening at the same time. It's not like one has to happen before the next, right? They're all just kind of happening at the same time. Okay, so that's prophase. And it looks more or less like this in real life. Okay, the next step of mitosis, right? So second step of mitosis is called metaphase. This is easy for me to remember because in metaphase, the chromosomes line up in the middle. Okay, so what's happening in metaphase is, remember that mitotic spindle? It moves the chromosomes to the center of the cell. I can't spell center. Center of the cell, right? And they're like single file. Now, 3-dimensionally, right? So the way they always show it in these pictures is they show like 1, 2, 3, 4, 5, 6, like they're all like lined up single file, like, well-behaved kindergartners. But in reality, it's a cell is 3-dimensional. So in reality, they're all in the middle, but it's not exactly a straight line. It's like this, okay? But they're all in the middle. And this is what that ends up looking like, right? Because they, can't keep their arms and legs to themselves. So they, right? So you can see them kind of, okay? That's metaphase, right? So we line up the chromosomes in the middle, okay? Now remember, why do we have 2 copies? Why did we duplicate our chromosomes way back in interphase? Why did we do that? Well, you're going to make new cells. Remember, you got to have complete copies of the instructions, right? So guess what's going to happen? I think you might be able to predict it. Our third step of mitosis is called anaphase. And what's happening in anaphase is now the mitotic spindle is separating the sister chromatids from each other, right? So the mitotic spindle is literally grabbing at the centromere, and each side of the cell is pulling one half. So we're unclipping our copies, right? We're separating the copies from each other, okay? So the mitotic spindle pulls, sister chromatids. Remember, those were just copies of each other, yeah? Apart. And because they're being dragged apart, they end up kind of looking like this shape, right? So if you're having a hard time wrapping your brain around that, take something that's long and skinny like a string or like a, you know, earbud cord. I don't know, whatever, right? You guys all have, I know you all have like AirPods and you're like, what kind of, you know, poor person is she that she has things with a string? It's called I will lose them because I have children. That's what that's about. Okay, so if you take a piece of yarn or string, right, and you put your finger on it and you drag it, it's going to make this kind of shape, assuming that it's something flexible, right? So as they're getting pulled apart from each other, it's like, no, don't take away my sister, right? And so this is what it actually looks like, right? As they're being pulled apart from each other. Okay. They don't really cry. They're not upset about being pulled apart. It's what happens. Okay. All of a sudden the pictures got different, right? So it was like, okay, these were all looking okay, consistent. Oh God, I don't like this actually. This really irritates me because now they're showing you the whole cell. And it's like, why weren't we looking at the whole cell before? I don't know. I don't know why they decided to do this, but I'm going to deal with it. Okay. All right. So the fourth and final step of mitosis, fourth step of mitosis is called telophase. Okay. If you're having a hard time remembering what order they go in, PMAT. Prophase, metaphase, anaphase, telophase, PMAT. Okay. All right. So what's happening in telophase? Okay. It's important to recall what happened in anaphase, right? So remember what happened in anaphase is we now had we separated those two copies into two separate piles, right? Yeah. So we've got our separate piles of DNA. Okay. And so now the other thing that might be helpful to remember is that after mitosis is done, we go right back into the cell cycle, right? So after the nucleus has been split in two, right? We split the DNA into two piles. Now things are basically going to go back to normal, except instead of one nucleus, now it's going to be two. Okay. So the way that I like to think about telophase is that telophase is essentially the opposite of prophase. But this is an important but for two nuclei, right? So we started with one nucleus and we're splitting the whole point of mitosis is splitting the nucleus in half. Okay, so that's what we're doing. All right. So what were the four things that happened in prophase? Four things. Now I'm making up steps. What were the three things that happened in prophase? Let's go back and look. So the three things were the nuclear envelope broke down. So what happens in telophase? Oh, we're going to reform that. Okay. What else happened? But for two nuclei instead of one. Yeah. Okay. What was another thing that happened in prophase? Well, the DNA condensed and it, went from that long skinny form to like the short fat X's. They never label this because it's probably not a word. Right? So the DNA, those strands of DNA, right, the chromosomes go back to chromatin, right? So they become those long, skinny strands again. Okay, I'm going to point out something else to you. Let's count the number of strands in each one of these new nuclei. 1, 2, 3, 4, 5, 6. That's the same, right? So our number of chromosomes has not changed. We started with 6, we end with 6. All we did was we separated identical copies from one another. Okay? All right. What was the other thing that happened in prophase? The mitotic spindle was formed, right? So do you see a mitotic spindle anymore? Not really. So mitotic spindle breaks down. Goes away. Okay, so that's telophase. So like I said, an easy way to kind of remember the events of telophase is to just think about we're reversing what happened in prophase. okay, but for 2 nuclei instead of just 1. Now, there's overlap between telophase and another thing that happens called cytokinesis. Okay, so the one important thing to remember about this is mitosis, remember, is all about the DNA separating. Cytokinesis is about the cell separating. I don't have enough room here. Cytokinesis is the cell separating. Okay, so see how there's not like a line, there's not like a bold line here between these two. The reason it kind of fades is that cytokinesis starts to happen at the end of mitosis and they overlap quite a bit. So usually cytokinesis is happening at the same time as telophase. That's why they made the picture different now, because they're trying to show you both of them at the same time. So, and showing cytokinesis too. Okay, that's what this business is about. Okay, so let's talk about cytokinesis for a hot minute. We'll come back to this. We'll come back to that too. So the way that cytokinesis happens is depends on if you have a cell wall or not, right? So if you don't have a cell wall, like our cells do not, the way that cytokinesis happens is you have a cell with those 2 new nuclei that we just produced, right? And ultimately, the rest of the cell is going to separate. So the cytoplasm is going to get divided between, right? And what happens is the development of something called a cleavage furrow. I know some of you are hearing the word cleavage and you're thinking of boobies. The word cleavage, to cleave, literally means to separate, to separate 2 things. And that's what cleavage is. It's a separation between 2 things, right? So a cleavage furrow forms, and what the cleavage furrow does is it basically makes it like pinches and pinches and pinches and pinches and pinches until you end up with 2 separate cells, okay? So this image must represent an animal cell, or at least somebody that doesn't have a cell wall, because it's experiencing cleavage, okay? In the case of plants, you can't, because they have this rigid wall, they can't do the like pinch, 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 pinch thing. So what happens in plants is actually a piece of cell wall starts to grow in the middle between those 2 new nuclei, and it grows from the center outward, and we call it a cell plate. Okay, so that's 2 different ways of doing cytokinesis, depending on if you have a cell wall or not. Okay, but remember, fundamentally, cytokinesis and telophase, while they happen at the same time, are technically describing different things. So telophase is describing what's happening with the DNA, the nuclei. Okay, cytokinesis is describing what's happening with the rest of the cell, but usually they happen at the same time. Okay, all right. Here's an image from a different textbook. Now, what I like about this is I like that it's separate and stepwise, and it's like we're going to look at one thing at a time here, right? What I like about this image is because it shows them together, you can kind of see the differences progress a little bit easier. So one of the things that I would recommend that you do is you just kind of like follow along, right? Looking at this image. Okay, 2 images, I guess, right? And think about the steps and just, right? It's another way of representing the same process. Okay, I'm not going to do it again because one of the beautiful things about videos is you can rewind. So if you're like, oh crap, I need to hear that all again, you can rewind and do this 8 million times if you want to. Okay, all right. One thing I will do though is let's do a little bit of practicing of name that phase. So first of all, we only talked about like 5 different phases. If we go back to our cell cycle, I can never aim correctly when I'm scrolling. I always over scroll, right? So we talked about interphase, and then we talked about the 4 different phases of mitosis. So we talked about 5 phases, technically, but this shows 6 pictures. That's because they copied one. They did one twice. I don't know why. I should take out the extra one, but I kind of like it there because it's kind of like, well, Okay, so let's pick this one. What phase is this? And how do we know? Well, I can see, so one way that this is different from this, I can see a cell and I can see another cell, okay, is look at the nucleus here. I can't see like distinct chromosomes in this. So since I can't see distinct chromosomes in this, that would be interphase. Okay. Now this one, I'm starting to be able to see distinct chromosomes. They look like little squiggles. But the fact of the matter is they're not arranged in any particular kind of a way yet. So it's prophase. All right. Now in this one, how are they arranged? Well, those dark squiggles are all lined up in the middle. Yeah. This is the repeat. That's also metaphase. Okay. How about this one? What's happening? Well, the chromosomes are getting, the two sister chromatids, right, are getting pulled apart. So what was that one? Okay. And then there are, remember, two things happening here. One of them is the nuclei are starting to reform. Yeah, so that's telophase. But also, you can see the cells are separating too. So it's not just telophase, but also cytokinesis. Okay, so that's what they look like in real life. All right. Okay, so mitosis, right? You start with some number of chromosomes. They're using 6 in your example. Those chromosomes are duplicated during interphase. Beautiful. So now we have duplications. Mitosis happens. You end up with 2 new cells. Guess what? Same number of chromosomes. Imagine that. Okay. All right. That's mitosis. I'm going to stop here. Then we're going to have a little video on cancer. Then we're going to do meiosis. Okay. So we're going to kind of break it up a little bit. All right. So that was mitosis.